time.
So good afternoon. So good afternoon, everybody. I think we are ready now to start the first session of this afternoon. And if everybody can sit, since we are already 5 minutes late. And now this session is really to try to introduce you to what IMI is already doing in the field of diabetes, and will be followed by another section on what is the future. And for this session, I would like please to invite representative of 5 IMI projects to please come and sit here. And here we have a representative of fairly different projects, and Werner Kramer will give you, is representing the Imedia project that is really on the more basics science of the biology of the beta cells. Then we have Harriet that will talk about the direct project that is more about patient stratification and type 2 diabetes. Then Birgit is a representative of the Summit project that is focusing on complication of diabetes, both for type 1 and type 2. Then we have Bart, that is a representative of the EMIF, the European Medical Informative Framework project, which is really a data-focused project with one important research pillar on metabolic complication of obesity. One of these is diabetes. And then we will have Elisabeth that is representative of a project in IMI that is called Europain, that is actually focused on chronic pain, because indeed also chronic pain is a bit problematic for diabetic patients. And now I would like just to give the voice to the representative of the project to give a brief introduction of what is the project, what the project has done so far, and what are still the challenges, on the basis, of course, of also what has been so eloquently said this morning by the patient in the room. So we can start maybe from Werner, let's say.
Yeah, Elisabetta, thank you very much, and it's a pleasure to be here and to give you a very short overview where the Imedia consortium is dealing with. So you all know that beta cell dysfunction or loss is a major trigger and factor for the initiation of diabetes and its progression to its complications, and it's by impairing normal metabolic control, as we heard this morning, not only glucose, but as well lipid and energy metabolism. Therefore, I think an ultimate or a major goal for better treatment of diabetes and maybe a route for cure of diabetes is the normalization of this impaired beta cell function in diabetic patients, either by restoring the function of available beta cells in diabetic patients, by restoring the number of beta cells, or as well in future on cell therapy by replacement. So the question is why we are not there in order to have therapies for that. And I think there are, there are a number of reasons which hamper a directed pharmaceutical research in this direction. On the one hand, we have a complete fragmented and scattered understanding about the factors, the mechanisms which finally lead to impairment of beta cell function and its loss. That's one point. And it has many factors. I think we have genetic factors, we have environmental factors, particularly stress and nutrition. On the other hand, we have fundamental experimental limitations in order to tackle it. The one is that we have not and will not have enough human beta cell material available for fundamental research. We as well, prior to Imedia, had no surrogate with regard to functional human beta cell lines. That has changed. In a minute I come back. A further barrier why we are not there where we want to be there is that we have a very strong biological differences in the beta cell biology between humans and rodents. And as a consequence, the experiments and results we obtain from rodents are only partially predictive for the human situation. And as a further consequence, we have not really the capability of a straightforward, directed search for drugs which improve beta cell function or restore beta cell function because we have these experimental limitations. And that's exactly what the mission and the goal of Imedia is. Imedia is now in 4.5 years running, and it's a collaborative effort between 12 leading academic groups in beta cell biology, 1 biotech company, and 8 major pharmaceutical companies committed to find new therapies for diabetes. The goals of Imedia have been defined very early. That is to generate or to deliver human beta cell lines, to elucidate in vivo imaging methods for beta cells, as well as to find predictive biomarkers for early diagnosis of beta cell misfunction or dysfunction and progression of beta cell loss. And in addition, to investigate the mechanisms underlying the loss and failure of beta cells under environmental factors like stress, oxidative stress, or nutrition. And the question, and I think it came up, is very clear. What is the impact, or what is the potential impact of the Imedia consortium for patients? And I would like to highlight this with 3 or 4 examples. The first, I think, major point was that within Imedia, we succeeded to generate the first human-derived cell lines which behave in most of the aspects like a human beta cell in an intact organism. And meanwhile, we are in the development of the 2nd and 3rd generation of these cell lines, which can be used for drug screening purposes. So in other words, the impact of this finding of the first human beta cell lines is that for the first time, there are reliable protocols available in order to start searches, straightforward searches for compounds which aim to improve the function of beta cell or to restore them. That was so far not possible. We had to rely on mouse and rats with a very, very poor outcome for the human situation. So in other words, this beta cell line, what we have found is a conditio sine qua non for any feasible drug approaches for looking for compounds improving the beta cell function. The second example I would like to highlight is starting here from mouse studies. We have investigated the impact of a typical Western high-fat diet on the physiological behavior, development of diabetes, development of obesity in 6 mouse strains. So in a nutshell, these animals were analyzed, and with phenotypic analysis, with biochemical analysis and systems bioinformatics approach, several phenotypes, pathways, and genes were identified, which may play an important role for the beta cell function or its sensitivity to impairment. What is done is currently underway is that by this approach, we could identify certain families of lipids in the plasma of these mice which correlate clearly to outcomes and measurements of beta cell function and diabetes. And what's going on currently is that we are investigating this potential family of lipid molecules in the plasma in a cohort with ensembles from an observational studies over 9 years to see whether this is a potential indicator biomarker for early diagnosis of diabetes and as a biomarker for monitoring its progression. A third example is that, to our knowledge, within Imedia, currently the world's largest repository of human islet materials and beta cell materials was collected. It's not only collected, it is very important that the protocols for isolation were standardized and approved. So in other words, as well in the project with the beta cells, we have generated reliable and reproducible protocols, which is an absolute necessity in order to do any, let's say, drug search studies. And currently, these human beta cell analysis of the materials from healthy, from type 1, from type 2 diabetes are now put into comparison with similar analysis from mouse or from mice in order to identify those pathways and ways which seem to be common and therefore probably being promising and predictive for the human situation. In addition to that, for designing of transgenic animal models which have an importance and relevance for the human situation. And finally, within Imedia, we are elucidating different ways and ligands in order to monitor non-invasively and image non-invasively beta cells as a way in order later on to be in the position to monitor beta cell loss in patients, for example, which have received beta cell transplants, and in order to see whether compounds aiming to restore function of beta cells show a clinical efficacy. So in a nutshell, I think Imedia has put together, let's say, a similar step forward that a directed, straightforward search for compounds improving beta cell function is possible, that we have put a knowledge platform to understand human beta cell physiology better and a technical platform in order to be able to initiate drug discovery programs with a realistic probability of getting success. So that's in a nutshell overview about Imedia.
Thank you very much. I would like to ask Harriet.
Hello, my name's Harriet Teare. I am part of the DIRECT project, and I'm based at the University of Oxford. The DIRECT project is focused on type 2 diabetes, and as has been mentioned this morning, it's really looking at why some patients respond very differently through their disease than others. So we have a number of studies at this stage looking at patients that are fast progressors, so they go from being high risk through to being sort of fully fledged type 2 diabetes and the different treatments that they've had to work through, and those that actually can manage it very comfortably with diet and lifestyle. And we're looking at exactly why that happens, and also why some patients respond to certain treatments very well, but others not so much, and why there's this disparity amongst type 2 diabetes. And we're trying to find biomarkers that will help us to predict which groups different patients will fall into, to help tailor their treatment to their own needs, but also to find better treatments for them along the way. So that's a very brief overview. We've just finished our second year, so we've just come to the sort of end of the first round of recruitment to the various different studies. And we're at the very exciting stage where the data is coming in and we're now starting to look at it. And my background, actually, I'm a researcher in a team looking at the ethics, the law, and the regulation surrounding medical research, rather than being in the lab myself. So we're very interested in engaging with our participants about some of the issues relating to how they are interacting with the project, what their views of some of the issues that we're tackling are, whether different patients have different ideas about personalised medicine and how that's going to affect them. And we're looking to do a large survey across our participants, but also more widely, to collect some of these ideas in. And we're also investigating different ways to actually tell patients about the research that we're doing and how it's progressing. So we have a Facebook page and we have a Twitter account, and we're trying to build up this presence so that patients actually know what it is that we're working on and try and have that engagement. And it's great to hear this morning all of the enthusiasm for that interaction. And if anyone has any tips for us on how best to engage, then we'd be very interested to hear it. Thank you.
Thank you very much. And now I would like to go to Birgit for the summit.
Yeah, thank you. And thank you for inviting me to talk about the Summit Consortium today. Summit is a consortium that is doing research on diabetic complications, and that is really based on the strong need to develop new medications which enable us to treat diabetic complications despite the mainly glucose-lowering drugs that we have currently available. The overall objective of Summit, which is now in its final year of research, so we have started year number 5, the overall objective of Summit is to develop new tools and methods that can be used in clinical trials, and by using those new tools and methods to make those clinical trials shorter and much more focused. Now, we've touched on that already this morning a little bit, but we are all very well aware that a diabetic patient at one point in time has a very high risk to develop complications. Those complications can occur in the small vessel system, which we then call the microvascular complications, which could affect eye vision, but also the proper kidney function. But they can also occur in the large vessel system, which we then call macrovascular complications. And that results in an increased risk that a diabetic patient has to develop a stroke or a heart attack. We also know that not all patients are at equal risk to develop complications, and that is the interesting thing that we would like to understand a little bit better. We also know that there are patients who are progressing really, really fast, whereas others do seem to maintain their condition over many, many years until they progress. So the goal of the biomarkers that Summit is trying to identify, which could be either genetic markers or any circulating marker in the body, should help us to identify those patients who are at risk to develop complications, but also to differentiate between the fast and slow progressors. If we had those biomarkers available, we could also use them in our standard medical practice to monitor the progression of a disease, but also, a potential regression if a treatment will work. And if we look into that from an industry perspective, then the other important component for us is that the way how we are currently doing our clinical trials when we investigate drugs on diabetic complications those trials are not only very, very costly, but they are also having extremely long duration. And many times we are not successful in developing a compound in that regard. So if we had biomarkers available, we could change this in a way that those trials would become much, much shorter, as we do not need to wait until the clinical endpoint is being reached. Those biomarkers would indicate us much, much earlier if a compound will likely work and if a patient responds to this treatment. So ultimately, I think the benefit, and we are talking about patients today, the immediate benefit that if Summit results are showing to be successful would be that we could not only speed up the drug development process and making hopefully better medications available in much, much faster manner, but we would also have biomarkers available which would help us to identify those patients at risk, and if we had a treatment opportunity to start this treatment early in the hope that we can then either slow down the onset of complications or in the best case prevent it. That is, I think, the dream of all of us. Now, talking about the achievements that Summit has can show up until today, then I think it's first of all important to mention that Summit is a really big consortium. We are having 26 partners, 6 industry partners, 19 academic partners, and 1 small biotech company. I've mentioned that we are in our last year of research. We have a little bit of an extension from IMI to complete some of the activities that will not end when we reach our official end date. But we are mainly interested in understanding the implications on cardiovascular disease in type 2 diabetes, but also microvascular complications with regard to eye disease in type 1 and type 2, and also kidney disease in type 1 and type 2. So one of our key achievements is, without doubt, to say, I believe, is that we have performed the largest ever made genetic discovery effort in the field of diabetic nephropathy, and we have analyzed more than 10,000 genomes, pooled that together with many other genomes that we had access to, and we have been able to identify one genome-wide significant signal which is associated with the development of nephropathy in type 2 diabetes. Those data are currently being integrated into other cohorts outside of Summit, and the hits are being replicated, so we need to wait to see what these actually means for us in terms of clinical findings. The second key achievement is what we could confirm, and that has been described already before, is a significant gene that is associated with the development of cardiovascular arterial disease and type 2 diabetes. And we have also seen some very interesting close to significant signals in the field of retinopathy. So those, as I said, those results are currently being integrated into other cohorts outside of Summit. We need more data in order to prove the validity of the results. And I think one of the biggest challenges that has been for us, especially in the first 2 years, was really to understand and get access to cohorts, to patient samples, to agree on the entry criteria, so the patient population we would like to investigate further, and then also to analyze, more than 50,000 genomes and other samples that we have looked into up until today. Summit is, and I would also like to highlight that because it's a really nice outcome. Summit is not only looking into genetic markers and circulating biomarkers. We are also trying to develop non-invasive technologies which we can use in order to detect complications. And there's one very nice result that has been mainly driven by Professor Jan Nielsen and his team in Lund University in Sweden. He has been able to develop an ultrasound device which is able to detect dangerous plaques. We see plaques related to atherosclerosis. Not all plaques are dangerous, but the ones who are dangerous can break up, and the resulting blood clot can cause a stroke or a heart attack. This result is a very simple, easy-to-use result. It's safe, it's not invasive, and by using it, we can actually identify which plaque is dangerous, who of those patients is likely at an imminent risk to develop stroke or heart attack. And they could, after this diagnosis, could then be referred to surgery. So we hope this device and the software behind it is currently being validated in a couple of European centers. We very much hope that this method can be taken up further and maybe at one point in time integrated into standard medical practice as a very powerful diagnosing tool that we had in order to diagnose those patients that are at imminent risk to get stroke or heart attack. Many things, many activities that we are doing, I think, and also when I talk with my immediate colleagues in the Summit Consortium, we all agree that none of us would have been able to do that by themselves. So it really requires a joint effort from a big and large-scale consortium. Okay.
Thank you very much, Birgit. And I would like to pass the word to Bart, talk about EMIF.
Thank you, Elisabetta, and thank you also on the organizers for inviting me here. It's been a wonderful experience to listen to the people for whom it really cares. I mean, why are we doing it? We're really doing it for the people here in the room. I mean, that's That's why we are working diligently. So I'm here to represent EMIF as a consortium, and as Elisabetta said, we are a little bit an odd one out maybe in this audience here, because we do not deal exclusively or primarily with diabetes. And if we deal with diabetes, it's type 2 diabetes. So what do we do in fact? Well, EMIF is a really large consortium. I'm sorry, Birgit. We have 56 partners, and I'm the coordinator. When I started out, I had a full set of hair, black, nice, shiny hair. Now look at me now. I'm still beautiful, but the rest. So we started out last year, can you believe that? Only 1 year ago. So what are we focusing on? Our objective is really to reutilize existing data. I mean, my colleagues here are talking about the creation of new data, about the creation of new studies to go deeper and to study more aspects of the disease or how the disease is starting. So we are trying to do this from the other way. So we are looking at what is already existing and how can we take full advantage of data which is already out there. That's really the objective of EMIF as a project. So we're looking at how to create a framework, and the framework is a combination of technical solutions, because when you deal with data, there are lots of process steps and a lot of handling of data involved on the one hand, but also, and that's probably our main challenge, how can we ensure that the reuse of such existing data is happening according to all privacy and legal and ethical aspects. So the whole social and procedural governance aspects around it. So, and the long-term objective with EMIF is really to create such an environment whereby data, which is already existing, would become faster available for researchers that are looking for large amounts of data, because there is a lot of data out there. So EMIF is really looking at creating the right circumstances to bring this data together and to increase the power with which we can actually do the research. Now, when I say data, and that's also one of the reasons why we have so many partners within EMIF, many of them are data owners, are participating in the project because they have access to data. The one topic is to create this framework, but to make sure that we don't do this kind of in an ivory tower type of mode or in an abstract way. We have also launched 2 research projects at the same time as starting the project. And one research topic deals with Alzheimer's disease, so dementia. The other one deals with the complications of obesity. So we're not starting from diabetes per se, we're starting from people with obesity. And as you obviously know, I mean, both dementia as well as obesity are 2 very important societal problems. And we're looking at how can we indeed collaborate with all of these data owners. Now, we started last year. Some of the achievements to date, given the fact that we have so much data, the first thing that we wanted to do was to really make an inventory, if you like, of the available data. And we did the kind of fingerprinting, if you want, about the data. So now we really know a lot of details about all of the data that we have. And the data is of very, very different types. It ranges from data that is created when we all go visit our GPs, our primary care physician. It's hospital data. It's data which is submitted to payers and insurers, so claims data. So that's let's say, the population-based data set. On the other side of the project, we also have access to a lot of highly characterized, very rich research cohort data. But those 2 data types are completely different. I mean, on the one hand, you have data that is 5 centimeters wide, so very few subjects, very few people in the database, but it's 10 kilometers deep because we know everything. And on the other side, we have data that is 10 kilometers wide on 52 million people across 7 European countries, but that data is maybe 5 centimeters deep. And actually, looking at opportunities to link these different data sets is one of the most challenging elements within the project. And I strongly believe that that is possible and that is a value. So when looking now more specifically at the research topic that deals with metabolic complications of obesity, what we try to do there is to identify markers that will kind of identify which people are at risk to develop type 2 diabetes, cardiovascular, but also some of the cancers that actually come with obesity. So hopefully, if the project is successful, we will help to advance the knowledge into how the disease originates and to also work on more prevention, because type 2 diabetes often can be prevented. I mean, other than type 1 diabetes, type 2 diabetes, there is something you can do about. And if people can be identified early on as being at risk to develop type 2 diabetes, then hopefully it doesn't get to the point where the diabetes actually breaks through. It's also interesting because not every obese person necessarily develops diabetes. Reversely, not every type 2 diabetes person is an obese person. So what is in there in the characteristics of these people that actually either prevents the disease to start or actually predisposes people to develop the disease? And that's one of the things we really like to study. The other one is in Alzheimer's disease, and I have to tell you that the basic researchers are completely thrilled. They are so excited. It's like small children, really. They are so excited to be in one project together between Alzheimer's, dementia, and diabetes, because there is a common pathophysiological link between the two diseases, and that's one side project that we're trying to evaluate. So in the long run, what do we hope will be the benefit of this project? I think that we hope to indeed support or work on two big societal problems, obesity on one hand, dementia on the other hand, and we really hope to identify markers with which we can identify people early on in the progression of the disease so that we can also work in a preventative way. What is the biggest challenge? I think it was already mentioned by Harriet. When you talk about reuse of data, well, you have to ensure that everything happens along the right privacy, ethical, and legal aspects. And so we're very concerned about how we can do that.
Thank you very much, Bart. And I'd like to give the word now to Elisabeth for the Europain.
Hello, everybody. I'm going to be telling you a little bit about Europain. Europain was set up around 5 or so years ago, and it's an IMI initiative in chronic pain. So the focus is not, like EMIF, on diabetes, but inevitably, diabetes being the disease it is, it impacts many sorts of morbidities, and that's where we're involved in a small way. Europain has around a dozen academic and a dozen industry partners all across Europe. We have 6 or 7 work packages, ranging from looking at genetic markers, preclinical models, healthy subject models. This is all of pain of course, and looking at patients and the epidemiology and development of pain following certain surgical procedures, and how almost psychological aspects affect pain. We all think we know what pain is. We all have suffered pain unless we have a very rare genetic disorder. We cut ourselves, we have a burn, we have a broken bone, we have surgery, and we know what pain feels like. But that pain sort of protects us. It helps the body heal. What if, which happens with chronic pain patients across Europe and the world, of course, around 10% of the population, that pain or some pain with no known cause is with you day and night for months and years of your life. You can imagine the impact that has on quality of life. Treatments, drugs, are not very effective in the chronic situation. Either they don't work for many patients, or they have horrible side effects. So Europain has a big job to do. Where do we link with diabetes? Well, One of the most difficult pains to understand is neuropathic pain. And the figures we have in Europain suggest that around 90% of patients or people suffering with neuropathic pain are those who have painful diabetic neuropathy. Therefore, this is the link. And in work package 2 of Europain, led by Laura Corradini of Boehringer Ingelheim and the teams in London and in Germany, they have been looking very carefully at preclinical models. Now, this is a sensitive area because people often don't like to think about animals being used in research. But unfortunately, when we're thinking about things like pain, there are no biomarkers for pain. Pain is what a patient, a person, feels. It is a patient-reported outcome immediately, but there are no markers to measure it. In Europain, we have mixed feelings about biomarkers for pain. Does it mean that if a patient or a person who doesn't have the biomarker doesn't have pain? Clearly not. So it impinges on philosophy as well. So to go back to work package 2 and our models, we've been looking at 3 different animal models so that we can test drugs for pain in painful diabetic neuropathy situation, but of course looking preclinically with the rodent models that we have. We look at face validity, construct validity, and predictive validity. Now that's very technical. What it means is, does it look like a diabetes type condition that the rodents have? Does it act in the same sort of developmental way? And can the drugs, which do sometimes help patients, also reduce the apparent pain seen in the models? So this is where we're really focusing on working for diabetes patients and pain. We didn't hear much about pain this morning, but we did hear about nerve damage. And I think the figures quoted by my colleague in Sanofi, in fact, Philip, suggested around 8 to 11% of patients who have diabetes will have nerve damage in some way. In Europain, we think it's a bit more than that. But so we would say probably between 10 and 20% of patients with diabetes will have a neuropathic and potentially painful condition to live with in addition to their diabetes and all the other morbidities. So where have we been successful? In terms of the diabetes situation, the models have been evaluated well, and we believe we're in a position to recommend one over the others. Now this will help us and maybe other groups as well, and clearly here we overlap with Summit, with what you were hearing just now. And we will be publishing a lot of these data, and these will be up in the public domain in scientific conferences, of course. And 3 papers have just been submitted on this work to the Society for Neuroscience, which will be in the US fairly soon. I think it's the US, might be Montreal. But is there more to do? Yes, of course there is more to do. At the moment, the aim of developing this model is to test potential new treatments to reduce pain. What we would really like to go on and look at, and I say we, I don't just mean Europain, is modify that disease process. This is an even bigger challenge, and this again is where we link, I guess, with the other EMIF and Imedia as well. And it shows the transverse nature of Europain, but also IMI across all these different work streams and work packages. So thank you for listening. I'm a little bit of an oddity in that I'm not one of your diabetes experts at all, but you can see that even in pain, we have the interests of diabetes patients at heart too.
Thank you very much. And now we can open for discussion. And I just would like, please, to ask to everybody that ask a question, please, to introduce him or herself so that we know who is making the question. And maybe all of you like to start.
Yes, so perhaps waiting for the other questions. One thing I would speak to Werner, Harriet, and Birgit, representing what we call the diabetic platform. And now the projects are well advanced. You arrive and you see the 1 year, the last year. What are the lessons we can draw on the patient perspective, and what would be the challenge you would see for the next step, which is IMI2? What is the key point, each of you, in your study? Now it's quite at the end. in the patient view on your side? And perhaps after we will have some questions from the audience.
I think one of the things that has been interesting for us is that the project has a number of different parts and is spread across a number of different countries with a number of different study centres. And so making sure that we're catering, or we're sort of thinking about all of those patients rather than just being UK focused or being sort of in a specific area has been quite a challenge, and making sure that language barriers and all of those sorts of things are thought about as well. But also, we've had the initial studies that we're doing at the moment, and we will have clinical trials, hopefully, towards the end of the project. So we want to engage with patients on both of those things, and making sure that the patients come back and keep engaged in order that the data that we're collecting is really powerful. So we've got a number of sort of different things that we're looking at that will be fundamental for the project's success.
Yeah, I think from our perspective, what impresses me personally most was the strong collaborative effort of the Imedia consortium. In other words, when it started, you know, all the companies were or are competitors concerning their markets. Nevertheless, it turned out that the, let's say, scientific challenges in order to get forward are so high that it really turned out to be one scientific group. No differences between the academic and the companies, and sharing data to an extent I never ever would have foreseen earlier. So, in other words, scientists from one company visit scientists in the lab at the other company. So why is this important? It is important, as I tried to outline, in order to get comparable protocols, comparable data and models. And one of the lessons learned was that, of course, every year several papers were published concerning a beta cell line. Basically, there was something, but not really reflecting anything for the human relevance. So in other words, what I think is, in order to tackle these really complex problems, it will not be possible, in my view, that one company alone or one institution alone will have the expertise, the power, the endurance to do it. I think, you know, this buzzword of pre-competitive, I personally see this much, much broader. Only with this collaboration, I think we can go further on. And I think we have some examples which may serve now as a rational basis in order to go into some therapeutic options investigations which never ever would have been possible earlier. And that is one point. And coming to the challenges, one of the challenges surely is these first consortia will come to an end in about a year or two. And I think what is important, and of course different teams have started to think about how we can, let's say, maintain the knowledge which is already elaborated and transform this to a stage where it can be of use for future collaborations. Otherwise, you know, it would not be a really optimal way in order to say, well, this is 5 or 6 years, that was a fine time. I think it is for medical progress necessary to harvest many of the treasures which have not yet been harvested. It just has begun, and I think this is one of the administratory challenges we have.
Yeah, and maybe if I bring that in relation to Summit, then I think what is clear and what we very much would like to see happening in the future is that we are moving from treating a population where some respond and some do not respond to treating a targeted population where we know that each of the patients who receive the treatment responds. In order to make that happening, we need to have those biomarkers. We can only identify those biomarkers if we had really, really big biobanks. So we need to get access to samples not only across Europe but also around the globe. So prospective studies, you know, the setup of biobanks in the UK is working hard on that, I think are an incredibly valuable source in future in order to make these findings happening. So your input, your participation, I think is really critical in future.
Okay, so now I would like really to ask the speaker of this morning, as voice of the patient, really, now you heard about this IMI initiatives, so considering what you expressed so eloquently this morning about the need to the patient, what is your impression? What would you think that you have heard that more have touched and you think is more near to your need? And of course, what could be developed more if you have any input for the future that would be most viable to build on what is already done? And I hope. Okay. Can we have a microphone there, please?
I could raise the question to all of you, but your projects are finishing. One is finishing in, I think, October 2014. If I look at, what was it say, Europain. If you should communicate to all the patients here in the room, you have to communicate. What would you communicate about your results for us? So this morning someone said, what's the benefit for us? What's in it for us? You all had a very nice story, and I may say that I can understand some research terms, so I can follow you anyway on the stage. But imagine that I'm a patient, I will now say patient with diabetes, and what's in it for me? Or for us. And for example, I'm very good in names. You said for the results or for the future that we need you. Did you need us before you started? Did you thought about us before you started? Because that was also one of the issues that was raised this morning. So I don't know who's going to answer something, but what's in it for us?
Yeah, I can try to answer to your question. Summit could not have happened if we had no access to biobanks. So all the data that we have used and analyzed, all the samples were already existing. And that was only due to the fact that we had really extremely good academic and industry partners who were able to access those cohorts and bring them into the consortium. In future, we want to do this prospectively. So we want to have new samples. We need to validate it in huge cohorts in order to identify what it actually means. To identify a biomarker, to identify your individual risk of developing complications. So that is, I know, a little bit of future at the moment because we have not yet reached this stage where we can actually implement this biomarker or any biomarker into the clinical diagnosing kit. But we have some tangible results, like, for example, the ultrasound device, which I think is a very nice finding and which might give you a little bit of a flavor of what might be a good outcome of a summit consortium and maybe also of other consortia.
I think for Imedia, that's my personal view. It's quite, let's say, clear what is the outcome. I think with the, I would not say luck, it was hard work, but with the availability of the human beta cell line, for the first time we can experimentally approach the factors which determine the decline of the disease. With that, we as well can initiate two things. Creation of animal models, of humanized animal models, which have a relevance for the human disease. Coming away from, let's say, playing around with a mouse, which has very, very poor importance, but you know, only afterwards. And the other point, very practical perspective, is that you can, with the methodologies of the pharmaceutical industry, start to begin searching and screening of large chemical libraries for compounds which improve the function of the beta cell. So far, it was not possible because you have no guinea pig, no cell line, and you have no animal model. So that is something which I see as a start point. On the other hand, as well, the knowledge, the availability of human cell line as well is a key step, in my opinion, in order to investigate the factors influencing proliferation of beta cells as a prerequisite for everybody is talking about stem cells. But, you know, it's a little bit neglected. What happens if you transplant the cell lines? What happens in situ? Where do they go? So you need some imaging experiments. So in other words, I think we see light at the end of the tunnel, or in other words, I think some of the ultimate goals for treating and maybe cure diabetes by repairing of impaired beta cell function by whatever means can for the first time be approached from a pharmaceutical perspective. You know, but of course we will not have the solution tomorrow, but today we are lacking some basic instruments measuring beta cell function and mass in vivo. We have nothing in hand. And I think this is on the way. And maybe a last point, what I tried to highlight is that we started from some animal investigations, identified some signatures of potential biomolecules to be detected in plasma, and in a translational approach are currently looking whether these signatures which correlate to readouts characteristic for diabetes have any significance for humans. If yes, the consequence would be we can probably these develop further as an early diagnostic methodology. If negative, we have the answer, okay, forget about this. In this aspect, the mouse and the man is different. This is second best outcome. So in other words, to make the funnel narrower in order to be able to concentrate on those scientific approaches which have a relevance for the human and not necessarily for the mouse.
Hello?
I think there's a mic.
No. Yeah, thank you. My name is Vannieuwenhuyse. I'm CEO of Biomodeling Systems. I would like to propose something. We are working in psychiatry, and the people designate, you know, the disease through the symptoms, and they have a problem. So we are working based on the mechanisms, and we describe, you know, continuum between these two diseases. What would be possible for diabetes to start from the autoimmunity point of view, and especially the loss of tolerance, and then to try to analyze, you know, these different autoimmunity diseases as a way to present, you know, this trouble. We think it's possible, and this could give us a way to reanalyze completely the diseases. And what was done in psychiatry, which has nothing to do, maybe could be done with the same principles in the autoimmunity. What do you think about this? Thank you.
Just a short comment from my side. I think this, if I understood correctly, this is a link to the type 1 diabetes, so the interaction of the immune system with the beta cell. That probably will be one of the topics to be approached with this foreseen first diabetes calls of the IMI2 initiatives. That's where we are. I think in July, the proposal will be more defined, but that, to my knowledge, will be surely something of interest in order to see, well, to what extent is type 1 diabetes from an immunological perspective similar to lupus or whatsoever, or are there, in order to elucidate the characteristics, and I think this is more than just, there are similarities, but discrepancies to other diseases. That's, I'm sure.
I have a slightly different question. 10 years ago, we were just patients. Now we are, for 98% of our time, our own healthcare professionals. What percentage will we be researchers in the coming 5 years?
I'd like to say that you're moving towards being partners already, and the understanding that all of the information and all of the expertise and all of the experience that participants have to bring into a research project is equally as important, if not more important, than the research that the researchers are carrying out in that project as well. So I wouldn't see that we'll move to the point where you're doing the research for yourselves, but that we actually have a much greater recognition and way to sort of deal with all of the information that the participants actually bring to the table already, and to make sure that we're capturing that as part of the research data as more of a conversation rather than as researchers are over here and research subjects are over here. And I think we're moving much closer together now.
Yeah, maybe trying to turn it a little bit around. Obviously, the question is how much percentage of your time would you like to be a researcher, but that's something else. But we are indeed now talking about this disease biology, so very, very deep molecular understanding how the disease originates. The question is about, okay, what about the patients, what about the people? And maybe within the EMIF project, I was mentioning that we are looking also at large-scale data sets. So again, we have about 52 million individuals from 7 European countries. Yes, we are only 1 year into the project, but if you would fast forward a little bit, my hope, my expectation for the project would be that we can actually take full advantage of the benefits of doing the EMIF project in Europe. And the benefits would be, in the end, I hope, also for the patients and for the society. What is the benefit of doing this EMIF project in Europe? Well, obviously not alone because we have IMI who is co-funding, but also because when you compare Europe with regards to patient-level data with the US, and within pharma we are typically fairly Anglo-Saxon oriented. When we talk about, and the newest hype, the newest buzzword now, in my domain at least, is real-world data. So data coming from EHR, from electronic health records, etc. So we're always looking at the Anglo-Saxon world, where in US you maybe have 150 million individuals in one database, so you can do whatever you like. Okay, that's cool. But Europe has two incredible benefits. And the two benefits of Europe are also challenges, at least one is. One is diversity. And if you think about how people are being treated, how diabetes is being treated, there is a lot of diversity across Europe. Treatment in the UK is very different from treatment in Italy, from treatment in France. Now, because we have that diversity inherently within the project, we can also look at what is more effective, which treatment pathway, what trajectory that a person is going through in the course of his or her disease, which treatment pathway is giving better outcomes, better results for optimal costs. So that's definitely a benefit that I hope that we can, together with the patients and with any stakeholder in the healthcare domain, can actually start to build insight into which treatment trajectories result in the best outcome. And with that insight, we should also be able to influence, together with regulators, together with authorities, how health policies are decided. So the diversity of Europe. Second, when, again, to compare Europe with US, in the US, people move on average, 20% of the population moves every year. So after 5 years, nobody lives in the same place. Okay, that's a bit exaggerated, but still, a lot of the people move around. So that means that people are in and out of databases continuously. So you don't have long follow-up. In Europe, by and large, we stay either within our locality, so we don't move as much, or we have national systems so that we can actually look at very long histories of patients. And when you study the progression of a disease like diabetes, well, you want to look at what happened 10 years ago, 15 years ago. So if you combine this longitudinal information with the diversity that we have in Europe, we should be able, if we're smart, to actually analyze and draw conclusions about which treatment options offer the best results.
Okay. Now we have one question there, and then Michelle. And then
okay, it's Robert Johnston from the European Patients Forum again. Forgive me, but it's really a follow-up on the previous question. I'm trying to hear from what you're saying how much patients are involved in really the whole process. So starting with determining the direction of research, actively participating in the research process, and then promoting the results after the research process and feeding through into the policy-making implications. You know, as EPF interacts with DG Sanko, we're actually starting to develop a very lively relationship now, where they're looking to us for new ideas and they're looking to us to help initiate their ideas. And that is working increasingly well. But I'm not really hearing concrete examples of that, forgive me, from the projects that I've heard outlined. I particularly like to see about the pain project, because that's something which is so you know, intimately related to the individual who's within that process. And just to finish by saying that, you know, we had 2 presentations this morning, but we had several presentations from patients, but particularly 2 young guys, you know, obviously very intelligent, very confident, and, you know, for heaven's sake, why shouldn't these people be being trained up, if they wish to be, and recruited to be active scientists and researchers? You know, I've seen in the UK, with this group I was talking about this morning, involve people with mental health problems being researched on, but becoming researchers themselves. Even people with learning difficulties starting to participate at the best level that they could with appropriate support. And I think, you know, amongst the patients, you have a huge resource. And I'm still not hearing that it's being used to its full potential.
Well, from pain perspective, thank you for the question, and it is a very important one. This is specific to pain, perhaps, but maybe other conditions as well. Pain suffered by patients is very different and a very different sort, so consequently is treated in very different ways in each country. So painful diabetic neuropathy may be treated by a diabetologist, maybe an endocrinologist, maybe a general practitioner, or maybe, if you're lucky, a pain specialist. How do patients and their carers and healthcare providers get together? It wasn't one of the work streams in Europain, but I can tell you that certainly an example I can tell you about is that of Chronic Pain Ireland, where all those people have been able to get together. Patients, of course, their carers and helpers, physiotherapists, alternative practitioners, nurses, physiotherapists, and pain consultants, and even regulators in that country have got together to try and give a voice to what is needed from the people who suffer. Pain, again, there is no badge that says, I suffer pain, that I mentioned before, no biomarker. So this also impedes a body of people coming together. There is no... there's a suspicion, I think, which still exists, that people who suffer pain are somehow complaining or malingering, and it isn't really there, which is absolutely not the case. Fibromyalgia we haven't mentioned at all. It's a very difficult chronic pain condition. pain, but not just pain, also muscle fatigue and gastrointestinal disturbances. So you have a very good point. One of the things I think Chris mentioned this morning was proposing involvement of people with diabetes, but it could just as well apply to any other condition in the design and setup and planning of early or late clinical trials for new medicines. And that's certainly something the industry, I think, would be very enthusiastic. Part of the problem about not doing that is the regulatory protection for anonymity of patients, so that there doesn't seem to be coercion. So there is a bit of a balance to be made there between patient desires and needs and potential coercion by interested parties, which presumably means the industry. Anybody else?
We have a mister here that was waiting already for a while.
Thank you. So one question, if you want to determine a good biomarker signature from this...
Can you please introduce yourself, please?
Oh, yes. Ulrich Giese from the Austrian Institute of Technology. If you want to get a reliable biomarker signature from these diverse samples from different biobanks, how did you manage it to get good annotation for every sample and have a comparable quality of the samples? That's the one question. And the second question is, it goes to Werner. So I think it's very good to have systems where you can test compounds, but I think at least it's as necessary to have systems to look for a side effect, at least in the context that a lot of anti-diabetics have been removed from the markets or have had a severe impact on their label on.
Quick answer, because I would like to have another couple of questions, and we have only a few minutes left. Thank you.
Okay, so I'm trying to be quick. I think I've mentioned that one of the challenges was, of course, that to harmonize the different cohort samples that we had available. So we really did an in-depth quality check before we've used them. Secondly, in order to bring it together, the work package that was about discovering genetic differences, we have used, whenever possible, the same patients if we had also samples, blood samples from them available, so that we could combine the findings, the genetic findings, with blood samples or urine samples at each of those individual patients. I think that was good, but there are still, I think, insufficiencies with regard to that we had not for everybody, where we had the genetic profiling, also the samples available in order to look for circulating markers. That's why we need to replicate that in other cohorts outside of Summit as well.
Okay, Uli, you are absolutely right. That is, I think, generic for every compound if we use this compound, let's say, systemically. So, but regarding beta cell restoration or functional improvement, we have nothing so far. So, but and if there is something, okay, in the whole discovery process, all these things have to be investigated in order to see what is the, let's say, long-term benefit versus adverse side effects. But one can think of as well other things that you can look for the compounds, and that is something which will become reality, looking for compounds which improve the beta cell or initiate a redifferentiation of precursors in vitro, which may, and these cells then, stimulated by a chemical compound, will then be the source for transplantation. So it's, I think it's in diverse direction. But prerequisite is that you have cellular and in vivo systems in hand which reflect to a high degree human, the beta cell behavior of a human cell. And this was so far not the case. So far it was, well, interesting papers with uncertain relevance for humans.
We have space for one more question.
It's not a question, it's a comment. And I would like to come back to the question that you raised. So just tell us what the consortium achieved, which is of direct relevance to patients' needs and expectations. So perhaps we will just still to wait a bit, because, you know, 3 years, 4 years in medical research, that's still very short. But we should be optimistic. I will just give you one example of the power of data sharing, of the power of the type of things that we were listening, especially to Imif, but obviously the others will also contribute. And this is an example from a project dealing with the development of new drugs for schizophrenia. So what has been done there within the New Meds Consortium is to pull data from 5 different companies. These were existing data, but never analyzed together. So the companies decide to make this effort. And they ask a third party, a neutral broker, to make the analysis. And what came out of it is of direct benefit to the patients. Why? By pulling the data, they were able to show that it's possible to reduce from 6 to 4 weeks the duration of clinical trial to demonstrate efficacy. They were able to show that it's possible to reduce by 30% the number of patients to be enrolled. And you can ask me, but how was this possible? So by analyzing the data, they show that what is needed to improve, the clinical trial is first to look at the gender balance, coming back to your point, to have a better balance between females and males enrolled in the trials. The second thing is to enroll patients at the very early stage of the disease, which is not surprising, but what clearly demonstrates. And the third point, which is also very relevant to patients' perspective, is to focus on certain types of symptoms. I will not go into details. So it means that indeed this type of collaborative efforts can really change things, both for those developing the drugs, but also for those who are waiting for the new drugs. And I think we should be optimistic from what we heard and, from the type of interaction. The people you are seeing, sitting together, probably that a few years from now, they would even don't know each other, really. And now they are talking to each other, they are working to each other. I would again emphasize the need to involve regulators very early on. And I think that with the example I gave you, that indeed you can be optimistic. You will have results which are of direct relevance to your expectations.
Okay. Thank you very much, Michel. And so now I think we can close the section, have a coffee break.